Thank you again, Vlad, for the presentation. And we will move to the next presentation, which is a joint presentation by Anton, Vlada, and Rodrigo. They will be talking about another machine learning analysis that we did and published recently. I actually don't know how you guys decided to do this. Are you going to start, Anton? Probably, yeah. Okay. I think you are muted for the time being, so please unmute yourself.
Okay, thank you very much for the kind introduction. And today I will present our more than 2.5 years efforts to study gene expression patterns between different mammalian species of various lifespan. And also applying some machine learning methods to find the maximum lifespan associated genes. So as Robbie mentioned in the very beginning of the workshop, we see tremendous differences between lifespan of different species. And there are many things that we can learn from long-living species. And we also know is that lifespan is also the trait that can have under evolutionary pressure. And that means that there are some traits called life history traits that can affect the evolutionary strategy of the animals and promote longer lifespan. For example, mass, so having less predators, ability to fly, gestation period, and so on. And for the traits that can be under evolutionary selection, we often see a phenomena called convergent evolution. And a good example of convergence is actually when totally unrelated species develop similar traits and body plans. Good example is aquatic species. And we also believe that to some extent it can be applied to longevity. So in our previous research, and spoiler alert, it will be presented by Mitya at 1435, we researched longevity of gray whale, and we compared it with 5 other species, and we found some interesting cross-species expression patterns, and it made us hopeful to actually try to get more data that will allow us to apply more sophisticated methods. So what you see here is our bioinformatic pipeline. I will also use it as a way to navigate our results. So we started from getting data and orthology annotations from ensemble and gene expression omnibus, and lifespan and life history trace data from NH database. And we used our own RNA-seq pipeline to process the data uniformly, and because of relying on ensemble, we had some limitations, so we couldn't include some long-living species, including, by the way, whales that were either not an ensemble or didn't have enough samples. So for the species selection, we selected 41 mammalian species for the 5-tissue model, and we processed 408 RNA-seq samples for the 5-tissue models, and then we applied 3 different types of models. We also looked in the very beginning because we were curious. We wanted to know if expression of different organs between species was more distinct or more similar than expression of the same organs between different species, and we found that actually same organ is more similar between species and different organs of the same species. Then we applied a bunch of models, and before us there were several papers, especially Gladishev's papers, that also applied different models to gene expression, but most of those models were linear models because we have small sample size, and also linear models are easier to explain. In our case, we started from linear models, but we also added non-linear tree-based models as well as causal models. So for the linear models, what was important for us is that we could build predictions on a per tissue level, and also make it for all life history traits, because linear models don't require a lot of data to train. Here is the chart showing how different genes and pathways behave in different organs. So blue is positive, red is negative. What you see is that actually gestation period and mitochondrial content correlates very well with maximum lifespan, and also some genes have consistent behavior in different organs. And in terms of pathways, we group the pathways in pathways related to longevity, pathways related to metabolism and immunity and inflammation, and other. And we found some classical longevity pathways like base excision repair, also apoptosis related pathways. In the same time, what was interesting, what we didn't find, was we didn't see any enrichments of mTOR signaling pathway. So our current hypothesis that maybe on an expression level, this pathway plays a higher role in terms of variability of lifespan in laboratory conditions, but probably between species and especially in wild condition, it can have a smaller role. We also got some results with ubiquinone and ubiquitin, well, and of course, as well as apoptosis that we saw in Wales paper. And we also found that some of the pathways that were enriched were mentioned only in several papers, like, for instance, metabolism of amino acids and unsaturated fatty acids metabolism was mostly mentioned in Gladyshev papers, but not very widely in other papers. And we also found some intersection with ionomics, inflammaging, and lipidomics study on sulfur metabolism, JAK-STAT signaling pathways, and PPAR signaling pathway. After that, we switched to nonlinear models, and the reason why nonlinear models were not widely used before is because, of course, they needed more training data, but also they were harder to explain. Luckily, due to new developments in explanatory machine learning, we were able to utilize Shapley additive explanation to get interoperable non-linear models. And here I give a word to Vlada Tyshchenko, who was our remote intern and contributed a lot to non-linear tree-based models. Please switch to Vlada.
Thanks, Anton, for introduction. I will start sharing my screen. We wanted to investigate nonlinear impacts of genes on maximum lifespan. And for that, we wanted to choose a powerful nonlinear machine learning model that would not only provide accurate predictions of the lifespan, but would be able to explain the impact of each gene on each sample's predictions and on predictions at the level of the whole data set overall. For that, we chose the combination of LightGBM and SHAP libraries, as they successfully can work together. LightGBM is based on gradient boosting trees algorithm, where the weak learner is the decision tree. And here is a small example of some trees that can be inside of this algorithm. So here we have a few trees. Each of them has its own set of features or expressions of some genes, and each produces its own output of the prediction of maximum lifespan. Then once we have predictions of by all of the trees, we can sum them up and have a final prediction. As for the SHAP library or shapely additive explanation, this is the explanatory model that is based on game theory, and it is able to output the impact of each gene on the or each feature on the prediction. So in this example, we have a base maximum lifespan value, let's say 0.1 years, and maybe this is the mean maximum lifespan in our data set. And then we can see how each gene contributes to the prediction of the maximum lifespan for each sample. Some of the genes would have positive impact, means that they would tend to increase the maximum lifespan predictions, like gene D, C, and A. And some of the genes would have a negative impact, or they would decrease the prediction. Then we would sum base value and impacts of all of the features or genes, having the final maximum lifespan prediction. And this final prediction should be equal to the output of the LightGBM model as they work together. We also had only 400 rows and more than 11,000 genes or features, which means that we had to have some powerful techniques of feature selections so that we would select only predictive genes from this huge amount of features. And we would need to avoid overfitting, as we have a much smaller number of samples than number of rows. For that, we had a few stages of feature selection. Each of the stages used LightGBM and SHAP selection. For the first stage, we would train 6 different models for each life history trait and choose only predictive genes for each of these 6 models. Then we combine the list, combine those genes into the single list, and this would give us 157 genes that would be predictive for any life history trait. But that was still a lot, so we had to create a second stage of the selection where the output would be only those 157 genes. And this model would try to find most predictive genes for maximum lifespan, so make it a smaller amount of set of genes. And this model, after the selection stage, outputted 57 genes. And as you can see, surprisingly had a better metrics of the accuracy of the model. And it can be explained by the fact that probably the first stage model had many noisy genes, as there were too many features of the model. And after the selection, we only left genes which were really predictive for the maximum lifespan. After the composite ranking with the linear models and Bayesian networks, we had the third stage, which would output only 6 genes. For each of the stages, we had a special cross-validation technique with multiple folds and multiple rounds of selection. For each model, we would select only genes that had non-zero SHAP value on some or all folds. And this would be this was different for different stages. We would also have stratified folds, means that they were stratified by maximum lifespan, and each test fold contained samples of 2 species that were not presented in the train folds. And this was done to prevent the overfitting by species column. So having this lots of rounds of selection, this gave us some stable list of most predictive genes. Here is the summary plot of 15 genes selected by the second stage model. And some of them had a significant mean absolute SHAP value, which was more than 1 year. And some of the genes were previously associated with aging, as we can see by their descriptions. We can also see that some of the genes had really high SHAP value for particular samples. And on this plot, dot represents 1 sample. And for example, DIRC4 gene had plus 15 years for the prediction of maximum lifespan for this particular sample. We can also see that, for example, for DIRC4, when it has higher levels of expression, it tends to have higher impact on lifespan. But once it has the lowest values, it has negative impact on lifespan, which makes this gene pro-maximum lifespan or positively correlated with maximum lifespan. We also know that genes can not only have individual effects on the lifespan prediction, but they might have synergistic, antagonistic, dependent relations. That's why we looked at the interactions between different genes, and this plot shows the heat map of topmost interacting genes. Let's look at the DIRC4 and RNH1 pair, which has one of the highest levels of interaction. And on the left side, we can see the same interaction, but on some other type of the plot produced by the SHAP library. And here we can see, again, here that each sample, each dot represents 1 sample. We can see that when the levels of expressions of DIRC4 are high, this cluster and this cluster, it tends to increase the magnitude of RNH1 impact on the lifespan. So here it's 2 or 3 years, and here it's -1 year. But once the expression of DIRC4 is low, so this huge cluster, it makes the magnitude or impact of RNH1 on lifespan really small or almost 0. So we can see some kind of nonlinear dependency and interaction between them. We also looked at the life history traits, and we tried to predict maximum lifespan using only life history traits to see which of them have highest SHAP value or impact on the predictions. And here we can see that mitochondrial content has the highest levels of SHAP value in it, and it is positively correlated with the maximum lifespan. Sometimes for some samples, it goes even higher than 40 years, the impact on the lifespan. And we also looked at the interactions between the life history traits. And here we can see the high level of interaction between mitochondrial content and body mass, for example, which was also seen before. So having this nonlinear analysis and some genes that we found that have nonlinear impact on lifespan, we still were not able to understand the causal relations between them, between genes and maximum lifespan. And now Rodrigo will tell how he was able to achieve it with his model.
Hi, I will share my screen. Here. Sure. Oh my. I'm sorry. I haven't trouble. Here it is. Sorry. No, this is not the screen. Here it is. Sorry. It's fantastic. Well, the objective here, as Vlad told, is to study the potential causal relationships between our target variable, maximum lifespan, and the genes, which that comprise our cross-species transcriptomic data. For this, in the first place, we need to understand this key concept. Maybe most of you have already heard about it, but I will do a quick overview. No, the concept is that of the Markov blanket, no, which given a graph that actually represents a probability distribution. The Markov blanket is the set of genes that that when fixed, when conditioned. I mean, when I have this target variable A, which is the maximum lifespan, for example, and if I condition this target variable on this particular set of genes, which is a fancy way to say that if I fix the value of these variables over here, this variable will be conditionally, of course, independent with respect to the rest of variables in the graph, which make them, strictly speaking, redundant. In a sense, all the information need are in this set of variables. And moreover, the parents, which are the ones which have this arrow that begins here and here, are called are the cousins, and these are called the spouses, and these are the children. To give such a to give or to give a causal interpretation to to a set of, to the relationship between, to a graph of variables, random variables, three assumptions must hold, and the algorithm, we thought, try to deal with the first two as best as possible, and the third one was a bit out of the control of a third one was a bit out of our hands. The causal marker assumption, there are a lot of nuances and specific things that and formal and specific things that one should explain in order to truly understand what's behind the definition. But in short, we can say that the causal marker assumption ensures that, given a graph, we can actually generate the data from the underlying distribution, and the faithfulness goes the other way around. Given the data of the underlying distribution, a sample, data that represents the underlying distribution, or a very representative empirical distribution, we can actually, through the correct methods, we can actually build a graph, a DAG, an oriented and a cyclic graph, an oriented and a cyclic graph. And causal sufficiency assumes that, for each variable, we already, our dataset includes all the confounders for each one of the variables within that dataset, which in our case, and in most of the cases, it's practical. It's really difficult to attain, and we only have transcriptomic data, which it's just part of the whole information that we should be using to explain our target variable, which is very high level. So this is a graphical depiction of the algorithm in order to basically what we're trying to do, as I said, is through is first of all to approximate our empirical distribution, which is our transcriptomic data, to the underlying distribution by using bootstrapping and to use a method which is variable constraint based Bayesian method called SES. Very similar to the MMPC. I'm sorry. MMPC. I'm sorry. The algorithm, which most of you already know, probably, and because this is actually more popular. And in the first place, we divide the original data set between the test set and the training set. We are going to save this for the last. And we do a stratified partition, by maximum lifespan, and then take a permutation, comprised of 4/5 of this training set, and save one for the validation. And we use this in the SES algorithm in order to find the parents and children of the Markov blanket. I mean, the parents and children are a subset of the Markov blanket. Basically, the causes and the effects of the maximum lifespan. And this is on one hand, and on the other hand, at the same time, if by using this data and this algorithm, we find, for example, that the parent-children set is comprised by five variables. Then we randomly pick 5 variables as well, from the same set. I mean, from our transcript from our data set, and we train a random forest regressor of 123. First with this this meaningful subset, and then with this meaningless subset on the validation set, and we save the RMSE of the. By the way, this is called for short a gene signature of maximum lifespan, and well, this I will call a random gene signature of maximum lifespan. Because it's random, and we save the RMSE of the results that our random forest regressor trained on the training data set gave us when predicting the values of the validation set. I mean, when using the validation set, and also the random counterpart. And we iterate 5 times for each permutation, and we do 2,000 permutations, which in total give us 10,000 simulations, and thus we get 10,000 gene signatures. The results, and because we are saving in each iteration, in each simulation, we are saving the signature we have found, and also their predictive power on the validation set. And and also of their random counterpart. So we calculate then the relative frequencies, which just mean the amount of time each gene appear in this set. For example, if a CEL, the gene CEL, appeared in 60% of all of the simulations, no, then the relative frequency will be 60%. And finally, we use a threshold to do the final selection of genes. And our data was originally highly normal, so we use a both side transformation called ORQ, order quantile, and we also find out that we can see that the mean is a function of the organ tissue. If we group them by organ tissue. So our first, we will first believe that a hierarchical Bayesian model will be a good choice in order to do the predictions. Nevertheless, because some of these groups have a very few samples, the results will be misleading. So a simple Bayesian model, or you want a simple linear regression, was used. And these were and we will leave this for one moment. No, we were here. Do you remember that we saved the RMSE of the gene signatures and the RMSE of the random signatures? This is the distribution of such saved values, and we can see that our model, the CNGD, which is the name I gave this algorithm, it's performing better than random in the training and in the validation set. No, the yellow ones are the random counterparts, and the meaningful ones are the ones selected by the CNGD are the blue ones. And these were the results among all the genes, among the thousands of genes that we were investigating. We did this selection, which is pretty robust because, for example, in NOXA 1, it was present in 98.99% of the gene signatures we found by using the algorithm, which is very notable. And then we have this one with 98, 93, 79, 64, and then it dropped. No, so these were the most robust ones. No, and using the and this is the most notable result of this algorithm, which is that the predictive power assessment. If we took the simple linear regressions and picked them, if we took the top 5, top 4, top 3, top 1 genes, and for each one of these lines we fit a simple linear regression on the training set, and then we test it on the test set, we get these results. No, which I mean, they have a behavior that we would expect, but the interesting thing is that there seems to be a positive correlation between the top number of genes selected and the generalization error. That means that if I start dropping genes from my linear regression models, the generalization error improves. which suggests us that well, we are doing a good job here, and by using the 50 carefully selected stratified partitions that Vlada explained in the previous section, these were the final selection of genes using the model I just explained, which are very similar, although not equal, to the ones found by just taking random simulations. And now my colleague, and this will be used. Like we will throw this in the consensus list pool. So, and that's exactly what my colleague Anton will explain next. Please go on, Anton.
Okay, I will summarize overall our results. As you saw from Vlada's and Rodrigo parts, is that we applied 3 different models to our data, and the most important part is to actually integrate our knowledge from those models. To some extent, we could use explanatory abilities of some models to explain results of another. For example, for linear models, we were able to compute models for each tissue. So we used it to explain tissue-wise behavior of causal and SHAP models. From SHAP models, we got Kendall-Tau correlations with longevity that we also use to explain the direction of influence of different genes. But overall, we wanted to get a nice short list of genes that we could look manually and find something interesting. And for this, we used very simple rank-based approach where we used some ranks from different models like relative frequency from causal models, absolute SHAP value rank from SHAP models, as well as number of repeats, how many times genes were selected in multiple repeats and folds in SHAP models, and as well as tissue linear rank. And we also took into account if the genes were also mentioned in GNH. In terms of GNH, we didn't see any significant enrichment of GNH genes in our results. That may suggest that maybe genes that are important for inside the model organisms for life extension may be between species, and also on expression level, they are not that important for life extension. So we got a ranked table, and then we wanted to find a threshold for a small number of genes that are the most explainable. And what we used, we built a so-called search stage SHAP-based model, and then we applied it to different top number of genes, and eventually we find that 6 top genes allow us to get more or less good results with Huber loss of 6.4 and R-square of 0.67. So we focused on 6 top genes and build the search stage model with them and used it more not for prediction but for explanation of their behavior. And what we see is that we have 4 pro-longevity genes, and NOXA and as well as carboxylester lipase that were, by the way, the most causal genes in our analysis. They have the largest influence. From NOXA, we know that it is enhancer for NADPH, which is one of the major reactive oxygen species sources. Carboxylester lipase has some indication about interaction with cholesterols and oxidized lipoproteins, but not well researched. For CalCoco2, we had a lot of information from linear models, and also for Speta20, it's also surprisingly uniformly behaved in different tissues for different models, and we have some aging-related hints for them in the literature. For anti-longevity genes, we have a little bit less information, but still there are some hints why they can be decreasing maximum lifespan. Also, we looked into sequences of the genes between species, and it didn't get into paper, but still I'm grateful to Elisa Martin to actually compare sequences, and we saw that most of those genes are highly conserved. That means probably they mostly modulate their influence through expression and also through different isoforms. So the isoforms were quite abundant. And also we found not only in these results, but overall, that many proteins express highly in testes. And for example, Speta20, even called spermatogenesis associated 20 factor. Surprisingly, in long-living species, it is expressed in a lot of tissues, and while in short-living, it is expressed primarily in testes, so it needs more research, but we suggest there may be an interesting mechanism for using protective genes for testes outside of testes for other tissues as well. And here are explanatory stage 3 model, and if you remember stage 2 models, you could see that in stage 2 models, some samples added a lot of years, while other samples didn't have a strong effect. For top 6 genes, we actually see very uniform influence, and also influence is strong in both sides. So low expression of Speta20, for example, significantly decreases maximum lifespan. That means that these genes behave in a more uniform way for maximum lifespan. We also build trajectories to see how they influence predictions in the model. We also see that mitochondrial GC content highly correlates with pro-MLS genes and negatively correlates with anti-MLS genes, and for some reason all genes are connected with highly predictive for body temperature in a positive way, and also we have strong dependence with gestation periods. That is also very important pro-MLS life history trait. And also pretty consistent expression for Speta20 and NOXA1 between different, and CalCoco2 between different tissues. So summarizing both results from stage 3 model as well as separate results from different models, we can conclude that in our analysis, we reviewed a lot of samples and tissues. I think it's an advantage that we use not only linear, but causal and tree-based explainable models. And we found that a small number of genes, like stage 3 genes models, can actually explain a big part of maximum lifespan variability. And we also found some patterns in between species and tissues and life history traits, like having metabolic related variables affecting lifespan in brain and quite independent lifespan affecting genes in liver. And we believe that we found potentially interesting genes to be further investigate for lifespan extension. So here are my gratitudes to the wonderful team that made it happen, and also to all people who helped us on the way. All the data, we had like 5 source code repositories for this paper, and I recommend to use the last 3 for reproducing our results. And hopefully we will also have a website to run stuff interactively. And with this, thank you for your attention. Sorry for being a bit late, and hope to answer some questions if you have any.
So thank you, Anton, Vlada, and Rodrigo, for a nice presentation. I know you guys know about caloric restriction, and because it's just before lunch, you probably decided that a shorter break is healthier for us. But if there is 1 or 2 urgent questions, we can take them, and we'll eat faster. Or yes, I see Vadim raising a hand, so he wants to ask something.
Anton, I have a short question to you and maybe your friends, if they like to answer. How do you evaluate the potential of machine learning analysis? Say, in a few years, maybe this approach will be more powerful than humans in searching the way for combating aging and so on. And maybe something is lacking, still lacking. So, your opinion.
Can I? Oh, please go on.
Well, I mean, maybe, so my opinion is what things that are lacking are, of course, more data, because despite we call omics as a big data, in fact, it's usually big in size, but it is very small in terms of number of samples. So here we didn't use any fancy deep learning things because our sample size was limited. So I believe that we need way more data for machine learning. As well, we need better representation, because the other problem of biological data sets is that we have too many features, too many species, too many gene products, and we need way more effective ways to represent it in a smaller dimensional space. And Rodrigo, please tell your opinion.
No, it was actually very similar to yours. I mean, we have the phenomena we are trying to describe is very complex, and it's just a matter of time. We use the more data we have. I mean, this could be very controversial and I think that pretty nuanced. In any case, the reason we use very statistical, I mean, more statistical methods than machine learning methods is that, or no, in short, it's just a matter of time. When we have enough data, machine learning methods will solve the problem for us. With us, maybe not all the problems, but many of the problems. In order to compensate the lack of data, we use a more statistical method.
I see. Thanks.
I kind of subscribe to their answer, so I don't feel the need to add anything. They are right. We will have more data, and the same methods will be applied, probably with even better results.
Okay, thanks.
Okay, are there any other short questions? If not, then perhaps we'll take a short break. So